Good afternoon. Welcome to Virology. We're going to build some viruses today. Actually, next time I'm going to bring some in, some models, not real viruses, because you have enough viruses on your own. You don't need mine. But the way viruses are built are according to very precise principles. So we're going to talk about that today. So virus particles all look very different, as you remember. And there's a reason for this. We can actually put a nice order to the way viruses look. So you can see here the many different kinds of particles. And at the end of today, you should be able to understand how each one of these is built. But they do have functions, right? They're not there to look good, although they look great, right? But they don't exist to be pretty. They exist for a variety of reasons, which includes to protect the genome, right? The genome inside is precious, the DNA or the RNA. This travels around the environment. It travels within organisms. It travels within the environment in water and dirt and so forth. And so the shell will protect it. Also, the shell's function is to make sure that the right nucleic acid gets into the particle, right? You can imagine in a virus-infected cell, there's lots of nucleic acids floating around. How do you make sure that the viral nucleic acid is the one that gets in? That's one of the functions of the viral structural proteins. And then some virus particles have envelopes or membranes. This one on the left here, that's influenza virus. It has a membrane around it. And so the viral proteins have to be able to interact with the membrane to get it to be part of the structure. Furthermore, they play a big role in getting the genome into the right place. The particles have to bind to receptors, a process we'll talk about in some detail on Wednesday. Then that binding typically leads to uncoding of the genome. Encoding means removal of the outer cover so that the nucleic acid is there in the cytosol or the nucleus, wherever it needs to go. For viruses with membranes, and these particles all have membranes, the way the nucleic acid gets out typically is by the membrane fuses with that of the host cell. And we'll see that on Wednesday as well. And then finally, in many cases, these structural components of the virus particle have to get the genome to the right place in the cell. The genome, of course, meaning the DNA or the RNA. So those are just a few of the functions of structural proteins of viruses. And before we go any further, we need to define some terms because they are, like many other things, unique to virology. So we'll start on the upper left, a subunit. When I say subunit, I mean a single folded polypeptide chain. You may call it a polypeptide in another context or a protein, but we call it a subunit of the virus particle. And then the next in the hierarchy is structural unit, the unit from which capsids or nucleocapsids are built. A structural unit is typically one or more subunits. So here, for example, on the upper right, outlined in black is a structural unit. And you can see it's made up of 3 subunits, 3 different colors of proteins. So that illustrates this idea. The subunit is the single folded polypeptide chain, and the structural unit is the unit from which nucleocapsids or capsids are built. Sometimes it's one protein, sometimes it's multiple, and sometimes it's multiple different proteins. It can vary, and we'll see those variations today. All right, so that's subunit and structural unit. Then we have capsid, which is Latin for box. And that's the protein shell surrounding the genome. All right, so it's drawn in the upper right, and we'll see many more of these today. All right, so that is a capsid. And those are for viruses where they're made just of protein. And finally, next envelope is the host cell-derived lipid bilayer that can be part of the virus particle. So these envelopes or membranes are always derived from the host cell because viruses do not make their own lipids. So it's a host cell-derived lipid bilayer. It's illustrated here with influenza virus. Now this virus on the upper right, that could have an envelope too. And we'll see lots of examples of that. So those protein capsids can be naked on their own, or they can have envelopes around them. And in fact, that's illustrated on the bottom right. This is a herpes virus where there is a capsid inside, as you can see, drawn slightly differently, but it's the same principle. The capsid has DNA in it, and that capsid is surrounded in turn by a membrane or an envelope, which is derived from the host cell. And this would be the infectious virus particle. I reserve a special word for infectious virus particles, and that is virion, as opposed to virus or virus particle. Virion means the infectious virus particle. Now, one term which confuses everyone every year, 50% of people every year, nucleocapsid. The nucleic acid protein assembly within the particle. We call it a nucleocapsid when it's a substructure, okay? So let's see. Here on the left is influenza virus. These are RNA protein complexes. And those are within a membrane. So these are nucleocapsids because they're a substructure. Whereas on the upper right, we have a capsid with some kind of nucleic acid in it. There's no substructure. And I'm going to show you some more pictures later, what would make that a nucleocapsid. This is a nucleocapsid down here on the lower right. It is a capsid containing DNA wrapped in a membrane. So the capsid is a substructure. I didn't make this up. It's the way virology is, but I want you to learn it. It's not that hard. It's when it's a substructure. A little bit of size here, just to put things into perspective. We use terms like nanometer, right? 10 to the minus 9th meters, which is the same as 10 angstroms, which is the same as 0.001 microns. We'll use these terms. I like to use nanometers a lot. So for example, an alpha helix is in protein, which is shown in the upper right. That's a nanometer in diameter. DNA, shown here, is 2 nanometers in diameter. A ribosome is 20. Poliovirus is 30 nanometers. And Pandora virus is 1,000 nanometers in length. And the biggest virus particles known are about 1,500 nanometers in length, okay? So those are the kinds of terms that we like to use. Now, a key point about viruses or virus particles is that they are metastable. Metastable, what does that mean? It means sometimes they're stable and sometimes they're not. Very simple. There are other meanings for meta, but we don't mean everything. So they have a function of protecting the genome. Right? So virus particles floating around in the environment protect, the shell protects the genome, so that's stable. But when they get into the cell, they have to come apart to release the genome, right? They can't be stable forever. So that's why they're metastable. Very simple, 2 states, very stable and unstable. And that's illustrated here for poliovirus, where actually binding the cell receptor is enough to trigger instability of the particle and release the genome. And Wednesday we'll talk about a number of different ways that virus particles become metastable. But the basic principles are as follows. Virus particles have not attained their minimum free energy conformation. So here's a graph of free energy G with time. And in order for a virus particle to come apart, it has to reach this minimum free energy conformation, which is shown in 3. But it exists in a higher energy state, number 1. And to get from 1 to 3, it has to go over an energy barrier, which is in number 2. So in other words, it's made a bit difficult to become unstable because you don't want virus particles coming apart all the time. So how does this work? When the particle is assembled, energy is put into it. We call it spring-loaded. And then that energy is released upon entry into the cell to allow the particle to overcome that energy barrier and get down to that low free energy conformation and basically come apart. So you use energy made when you assemble the particle to attain that. Now, how do you get metastability? This may seem like an impossible task, right? But not really. The stable structure, as we'll see in a few moments, is made by taking many identical proteins. To make a virus particle, you take many identical proteins and repeat them, and that gives you a symmetrical arrangement of proteins and maximum contact between them. So that kind of symmetry and arrangement gives you a stable structure. That's why virus particles are very stable. You can see this illustrated up here in the upper right. This virus has 3 capsid proteins. They're repeated many times, so that gives you a very stable structure. How do you make it unstable? Well, first of all, these are not covalent bonds that hold the proteins together. They are rarely. There's always an exception. There are some examples of covalent bonds, but most virus particles are the subunits, the structural units are joined non-covalently, and that allows you to take it apart to release or expose the genome. And again, we'll see next time all the different ways that you can take a virus particle apart. All right, the first quiz question. Viral capsids are metastable because A, they must protect the viral genome when the particle is outside of the cell. B, they must come apart and release the genome into a cell. C, they have not obtained a minimum free energy conformation. They are spring-loaded. All of the above. How did we do? The answer is all the above. Almost everyone got that. Every one of these is right. The capsids are metastable to protect the genome, right? Stable. Outside of the cell, they come apart. In the cell, that's unstable. They haven't reached that minimum free energy conformation, and they are spring-loaded, which means that the energy used during assembly is used for disassembly. I'm going to talk a lot about structures today. I just want to briefly talk about the tools of structural biology. How do you solve structures of things you can't see? There are a number of techniques shown here: electron microscopy, X-ray crystallography, cryo-electron microscopy, or cryo-EM, and cryo-electron tomography, nuclear magnetic resonance spectroscopy. And if you have the textbook, you can read a lot more about these in the textbook. That I am standing next to a cryo-EM machine up in Ithaca, New York at Cornell. Millions and millions of dollars for these things, but they're really useful, as you will see. Okay, the era of modern structural virology begins in 1940. We already encountered this publication where Helmuth Ruska used an electron microscope to take the first pictures of virus particles. And there's the paper in 1940. And you can see this is an E. coli on the right, to which are attached many E. coli bacteriophages. And so you can start to see a little bit about these particles, right? It looks like they have some kind of a head with symmetry, and it looks like there's a tail coming out of the head, attaching it to the bacterium. There are several problems with electron microscopy, though, and that is that biological materials don't have contrast. They have very little inherent contrast, so you have to stain them. And for electron microscopy, we do negative staining with an electron-dense material that will scatter electrons. Okay, so the electrons will not pass through it. It will scatter them, and so you're basically getting a negative outline of the virus particle. Compounds like uranyl acetate and phosphotungstate typically used to scatter electrons. The main problem is the resolution. Maximum resolution of electron microscopy is going to be about 50 to 75 angstrom. And remember, an alpha helix is 10 angstroms in diameter, so you're never going to get any detailed structural interpretation. But here on the bottom are some pictures, so you can begin to see what virus particles look like. Here, all the way on the left, is an adenovirus. And so you can see that the particle itself is negatively stained, right? It's coated with phosphotungstate, and it's reflecting the electrons, whereas the electrons are passing through the surrounding area and making the film black, essentially. And here's hepatitis B virus, influenza virus, and poliovirus. So you can get sort of a general idea of what virus particles look like, and many people did this for many years, but that's as far as you can go. So for example, for influenza virus, you can see, they're spherical. They seem to have these notches or something, which were eventually called spikes. That's where the word spike came from, but that's about it. You need more higher resolution approaches to know more. And next came X-ray crystallography, which is a very difficult technique, yet for viruses will get you 2 to 3 angstroms. So now we can look at the polypeptide chain and see how it's composed of. To do this, you need to make a crystal. You need to purify viruses and incubate them in some way so that they form crystals, like salt crystals. And that's very difficult in an empirical process, but if you're lucky, eventually you'll get crystals of viruses. And then you put them in an X-ray beam and you irradiate them, and the X-rays will bounce off the atoms in the crystal, and you'll be able to collect the bounces using a diffraction collector, a computerized detector, basically. And each of these dots will be information about where all of the atoms are located. This used to be done with X-ray film. You may remember the famous X-ray from 1953 from Rosalind Franklin, which showed the structure of DNA, which Watson and Crick stole off her desk to study. But that was an X-ray with a beautiful diffraction pattern of DNA. And it was very clear from that the nature of DNA structure. We don't use X-ray films anymore now. We use computerized detectors. But in fact, we now mostly use cryo-EM for viruses because it's much faster. You can get almost as good resolution, and you don't need to make crystals. That's the good part, because many people spent years of their life trying to make a crystal. They would make a crystal and put it in the beam, and then the crystal would explode. Can you imagine? So this is much better. Cryo-electron microscopy. Think of a CAT scan for viruses. So these three individuals, Dubouché, Frank, who's up at the medical center, and Henderson all figured out different aspects of cryo-EM to solve it. So what you do is, you don't have to stain anything. You take your virus particles at the top, you just freeze them. You have to freeze them in a certain way, but you freeze them to give them contrast. And that's what one of these three figured out, the conditions for freezing. Then the assumption is every virus particle is in a slightly different orientation, right? The way like a CAT scan, when it's rotating around you, is taking pictures in a different space. These are randomly spaced on the grid. So you take hundreds and hundreds of pictures, and then you digitize each one of them. And then you'd have to do some statistical manipulations. You do Fourier transformations, you determine the phase centers, and then you do a reverse Fourier to get the virus particle. I'm not going to pretend to tell you how that works, but the main key is you can synthesize all these different positions of the particles into a 3D reconstruction. So that's cryo-electron microscopy. Cryo-electron tomography, you take a single particle on a platform and you just tilt the platform in many different ways and take pictures. So you don't have to have a lot of particles present. You just take one particle on a platform and tilt it and take pictures as you're tilting it. So this is the Nobel Prize in Chemistry 2017. Really amazing. Amazing how quickly you can do things. So here's some examples. This is two structures of poliovirus. The first on the left is 2.9 angstroms. That's X-ray crystallography. So many years of work trying to figure out how to make crystals and diffract them and so forth. But the result is you know where every atom is located in the particle. And the result is a file. There's a place called the Protein Data Bank where you can go and download these files, and you get XYZ files for the three-dimensional location of every atom, and that's how you can make these pictures. And on the right is the cryo-electron version, 20 angstroms, much less resolution back then, but now it's equal to X-ray crystallography. And you can see, you can only see the surfaces of the virus particle. You can't see any details about atoms. But now cryo-EM has equaled X-ray crystallography, and it's also much faster. So here are a couple of examples. Here's Zika virus in 2015. Started to cause outbreaks of microcephaly in South America, so people started studying it. And so here is the three-dimensional structure by cryo-EM. And it's shown in two different ways to you. So on the upper left is a space-filling version where every atom is shown as a sphere. And you can see some symmetry here, which we'll talk about in a moment, right? You can see 1, 2, 3, 4, 5-fold axis of symmetry. You can see a 2-fold axis and a 3-fold axis. But then this version is a ribbon diagram where I'm just tracing the alpha carbon backbone. I've taken off all the side chains to make it clear. And I've displayed it as beta strands or alpha helices or just random coils. And so you can do this on your laptop. It's easy to do nowadays. And you can zoom in anywhere, you can select areas, you can do amazing things depending on what you're interested in. Now, back in February of 2020, a new virus was circulating, SARS-CoV-2, and Jason McClelland at the University of Texas, Austin, and his colleagues solved the structure of the spike within a month, 6 weeks of having the sequence. So that's the virus there on the left, an envelope virus with a number of glycoproteins in it. And the prominent one is the spike, which is a trimer. And so the sequence was released towards the end of December 2019, which was an interesting story in itself, but I won't go through it. He got the sequence like many other people did. He made the protein, and did the cryo-EM and solved the structure. So here's the spike protein. On the bottom is the part that's stuck in the virus particle. And then different parts of the spike are colored differently. In particular, the green is the receptor binding domain, and the blue is the N-terminal domain, among other things. We'll talk a lot about this in this course. So that's amazing that you could do this in 6 weeks. And then many people can have access to the data to do their experiments. So as soon as the variants arose, they could see exactly where the amino acid changes were and the implications for function. This is a different kind of view of SARS-CoV-2. This is the whole particle that has been reconstructed. So we have the spike protein, whose structure I just showed you, and then the nucleocapsid protein wrapped around the RNA in green is shown here, and then some other viral proteins in the membrane. So this was put together by a group of individuals from the individual cryo-EM structures. There doesn't seem to be a limit to how big structures you can make. There is sound here. It's the sound of waves. You can't hear it though. This is done by Shun Chow at UTEP, who spent many years solving the structure of this virus. This is a giant virus that infects a eukaryote that lives in the ocean. And it's a 300 nanometer particle. It has 15,000 capsid proteins, and it took 3 million CPU hours to solve the structure of that particle. He did it by cryo-EM. So it's really, really remarkable what you can do nowadays with computational biology. Now, we learned that there are 7 different kinds of genome. Today, we're going to learn that there are 3 different kinds of virus particles. So if you take the 3 or the 7 train, you'll never forget that. The 3 kinds of structures of virus particles are helical, icosahedral, and complex. Complex is kind of a cop-out because there's no symmetry involved. We don't really know how they're built. So I'm going to save them to the end for very little attention. But helical and icosahedral are really interesting, and we need to understand how they work. So you know that Watson and Crick solved the structure of DNA with Rosalind Franklin's help, but they actually also contributed a lot to structural virology. They noticed, so remember, this is the early 1950s now. People are taking pictures of virus particles by electron microscopy. They noticed that most particles were either spherical, like these in the lower right, or rod-shaped. Rod-shaped being like tobacco mosaic virus. And so they developed, and of course, if they had just waited 30 years, it would have changed their view. But since they were working in the 50s, that's what they had. And so they thought, well, maybe this means that virus particles are built using just a few proteins repeated many times. And that's why they have these specific shapes. They call this genetic economy. Nowadays, we know that we don't need to invoke genetic economy because there are giant viruses with huge genomes, right? But we didn't have any of those back then. And that's the way the idea evolved. And the idea is that for rod-shaped viruses, you have, again, identical protein subunits. The subunit, the very lowest level, the protein subunit, is repeated with helical symmetry. And for round viruses, those protein subunits are repeated with icosahedral symmetry. And what those means now, I would like to show you. What those mean, I will show you. So there are 2 rules for symmetry, and it explains how virus particles are built for the most part. First, each subunit, remember subunit, protein subunit, has identical binding contacts with its neighbors. So you're getting repetitive interaction of chemically complementary surfaces. That gives you a symmetrical arrangement. And two, these contacts are not covalent, something we've already said before. So you can get reversible, error-free assembly. So those are the rules for self-assembly of virus particles. And these rules are very useful practically, actually. It turns out that if you produce the capsid proteins of many viruses in cells or in extracts, they will self-assemble into a virus particle. We call them virus-like particles because they don't have genomes in them. But the vaccines for hepatitis B virus and the vaccines for human papillomaviruses are virus-like particles made in yeast. That is because the subunit, there's a single subunit for both of these viruses. All you have to do is make that protein, in this case in yeast, it folds and assembles into virus particles. All the information to make the virus particle is right there in the sequence. And so you can see you start with one protein and then you make intermediates. This one is called a pentamer. There are 5 copies of the subunit there. And then many of these pentamers form an empty particle. So these are the rules of symmetry in action to make vaccines. I think it's just amazing. All right, helical symmetry. We have a subunit, which we call a coat protein, and they're going to be interacting with each other and with the viral RNA. So here's an illustration of that. The coat protein subunit is shown yellow here, and if you put a bunch of those together, they form a helix, which is shown there. And this is the actual cryo-EM structure of tobacco mosaic virus. And you can barely see the individual coat proteins forming that helix. You can see coat protein subunit. And each coat protein is bound to the RNA, so it interacts with the RNA as well as with another coat protein. So that's helical symmetry. Essentially, the coat protein is making a helix of protein, and in the process is bound to the RNA. So that is actually tobacco mosaic virus, the first virus discovered. It's simply a helical structure with protein and RNA. Nothing more. Eukaryotic viruses also have helical symmetry. This one is a paramyxovirus, Sendai virus related to measles virus. Again, there's a coat protein. In this case, it's called a nucleoprotein that is interacting with the viral RNA repeatedly in a helical format. Interestingly, animal viruses with helical symmetry are always enveloped. The tobacco mosaic virus that I showed you is not enveloped. Plant helical viruses don't need to have envelopes. But animal viruses do. I don't know why, but you can see this electron micrograph of a helical animal virus. And actually, this one is broken. You can see the helical nucleocapsid spilling out of the particle. By the way, this is a perfect example of a nucleocapsid. So this virus is not a nucleocapsid. There's no substructure. It's simply the RNA bound to a coat protein. So we would call it a capsid. But if you wrap a membrane around it, now it becomes a nucleocapsid. This is the best example of what a nucleocapsid is. If the virus were just the naked coat protein RNA, it would just be a capsid. But because it's enveloped, it is a nucleocapsid. Okay, so that, in my opinion, is the best explanation of nucleocapsid. Rabies and vesicular stomatitis viruses also have helical symmetry. These are typically bullet-shaped viruses, very characteristic. As you can see in the diagram on the upper left, it's essentially an enveloped virus, and within it is a helical nucleocapsid, very much like the one I just showed you, but it has a pointed end and a flat end, which you can see in these cryo-electron micrographs. So here is the virus particle on the right. There's the nucleocapsid RNA in green and red. There's the cap at one end, and then it's flat at the other end. And then there's another protein layer on top, and then the membrane. But these coat proteins, I haven't shown you them individually. There's the nucleocapsid protein of vesicular stomatitis virus. It's a two-lobed protein with a groove in the middle, and that's where the RNA fits in that this nucleocapsid protein will be binding to. And then again, this is a cryo-EM structure of that complex. There are many envelope viruses with single-stranded negative sense RNA and helical capsids. The paramyxoviruses, which we've just shown, there's your helical nucleocapsid. I showed you a picture of one broken with the nucleocapsid coming out. The rhabdoviruses, influenza viruses, again, an envelope surrounding an RNA protein complex. The filoviruses, long filamentous viruses, but again, the RNA protein complex in the envelope is the nucleocapsid. So all of these are examples of nucleocapsid, the nucleic acid protein assembly that is packaged within the virus particle. So one more picture of nucleocapsids and not nucleocapsids, okay? So there's tobacco mosaic virus, coat protein and RNA. It's a capsid. It's not a nucleocapsid. Next to it, some virus with icosahedral shell, which we'll talk about right now, and some nucleic acid. It's a capsid. There's no substructure. Now below it, that virus, if you put an envelope on it, now the capsid becomes a nucleocapsid because it's a substructure. And the same with tobacco mosaic virus. If you put an envelope on it, now it's a nucleocapsid. Anything with a substructure. These two on the bottom are not nucleocapsids because they're just nucleic acids within an envelope. There's no substructure. So the nucleic acid on its own is not considered a substructure. It has to be protein nucleic acid assembly. Then we have a coronavirus with an RNA protein complex. It's a nucleocapsid. And then an unusual one here where we have an icosahedral particle containing a nucleocapsid. So those, I think, should cement for you the concept of nucleocapsid. And there are also plus sense RNA viruses which with helical capsids, famously the coronaviruses, right? SARS, MERS, SARS-CoV-2, et cetera. They have RNA genomes that are wrapped in protein within an envelope, so they are a nucleocapsid structure. By the way, when the virus was first isolated from the early patients, they took electron micrographs. You remember we talked about how they identified the receptor and they stained cells, and that we saw CPE caused by the BALF from the patient. They also took electron micrographs, and here's one of the first publications. You can barely see these spikes on the particle. So here's a classic photograph of coronaviruses. They're so called because of the halo of spikes resembling a solar corona. And so this new virus appeared to have a halo of spikes as well. DNA and RNA viruses can have helical symmetry. We've talked about tobacco mosaic virus, which is a plus strand rod-shaped virus, but they can be flexuous. They can be flexible. They're plus strand RNA viruses of plants that are flexible. They're single-stranded viruses of bacteria that are flexible. And here we have double-stranded DNA viruses of archaea, both flexible and rod-shaped. So all kinds of structures. These on their own would be capsids, with the exception of this one, which would be a nucleocapsid because it's a protein bound to the genome, which is DNA wrapped in an envelope. The rest of them are just capsids. Now, if you ever saw these buckyball these magnets, I forget what they call buckyballs. They used to sell them, and then they stopped because kids were swallowing them. and then now you can buy them again. I have thousands of them. I really want to make a big virus, but I haven't had a chance. You can really make nice viruses with them. This is a great helical virus that I built some years ago, and you can unwind it and get a sense for the principle. All right, our next question. Which of the following describes virus symmetry and self-assembly? A, the bonding contacts of subunits are usually covalent. B, the bonding contacts of subunits are usually not covalent. C, each subunit has different bonding contacts with its neighbors. D, self-assembly of particles does not occur. E, none of the above. Which describes virus symmetry and self-assembly? All right, let's have a look how we did. The bonding contacts are usually non-covalent. That's correct. They are not covalent. Each subunit has different contacts with its neighbors. No, the symmetry means they're all the same. All the interactions are the same. Self-assembly doesn't occur. You know that it does. I gave you nice examples for 2 different vaccines. So bonding contacts are usually non-covalent. Okay, round capsids. We finished with the long ones. How do you make a round capsid from proteins with irregular shapes? So there is poliovirus, lovely spherical particles, right? And the proteins that make it up look like this, decidedly not spherical. So how do you do this? Well, Watson and Crick were involved in this, and they present these 2 clues to answer this question. First, all round capsids have precise multiples of proteins. Multiples of 60 are common, like 60, 180, 240, 960. Actually, it's not true anymore, but back then it was true. And number 2, spherical viruses come in many sizes, but the capsid proteins are always about the same size, 20 to 60 kilodaltons. So in other words, as the capsids get bigger, the capsid proteins remain the same. So what's going on there? Well, they concluded that these particles are made with icosahedral symmetry. So what is that? Icosahedral is a platonic solid, one of multiple. This one is a solid with 20 faces. Each face is an equilateral triangle. 20 of those faces making up that solid. And as a consequence of the repetitive nature of each equilateral triangle, you get axes of symmetry, 5-fold, 3-fold, and 2-fold. They're 12 each. So for example, here's a 5-fold axis, and here we're looking at it down. All it means is that there are 5 equilateral triangles around that 5-fold axis. The 2-fold axis, there are 2, and the 3-fold axis is 3. So it turns out that an icosahedron lets you make a closed shell with the smallest number of identical subunits. You could make a virus with 60 copies of the same protein. That's what an icosahedron gets you. Plus, it's very stable. Of all the platonic solids, it's the most stable. Okay, so how do we make a virus particle out of this? The simplest ones are made up of 60 structural units made of one protein. Remember, that says structural units. That's one level higher than subunit, because they're only composed of 60 protein subunits. 60 protein subunits, so that means each structural unit is composed of only one subunit. So this means all the interactions are identical. All 60 of those protein subunits are interacting with each other identically. Now, these particles are actually not icosahedra. If you look at them in the EM, they're spherical, like here. But the point is that the proteins are arranged with icosahedral symmetry, with the repetitive rules governed by icosahedral symmetry. So remember that. They're not really geometric looking in the EM. So here's an example of a simple icosahedral virus. It's a virus made of 60 copies of one capsid protein. So 60 subunits, and the structural unit contains one subunit. And this is a parvovirus, which we've mentioned before. These are small single-stranded DNA containing viruses. And that's the capsid protein right there. And they have marked the axes of symmetry for you. And so 5, for example, here is the 5-fold axis of symmetry, which simply means there are 5 copies of this protein surrounding it. So 60 copies of that protein is enough to make up these small virus particles. Moving up, how do you make a bigger virus particle? As I said, you don't use bigger proteins. You just add more of them. That's the key. And so now when you add more proteins, you're changing the environment of those proteins. When we had 60 of the same protein, every area on the capsid was identical. But now that we have more, that's all changed. And now we have what are called pentamers and hexamers. So I'll show you, for example. So each of these commas is a protein. And we have in this model 180 identical protein subunits. So it's the same protein repeated 180 times. So all those commas are the same protein, but they're in different environments. For example, see this green pentamer, there are 5 copies of the protein around it, 1, 2, 3, 4, 5. But somewhere else on the particle, you will find 6 copies surrounding the triangles. Here, the purple, 1, 2, 3, 4, 5, 6. So you'll find pentamers and hexamers. That's simply a consequence of adding more subunits. When you do that, you have to disrupt the uniformity, and now you have pentamers and hexamers. And so as a consequence, the bonding environments are different. It have to be, right? If you have a pentamer, it's going to be different from a hexamer. So we no longer say that everything is equivalent, everything is identical in these particles. We can't, because it's not. We say it's quasi-equivalent. And this was an adaptation by Casper and Klug, 2 structural biologists working in the 1960s. They realized that with bigger particles, Watson and Crick were wrong. Everything is not identical, but it's close. And they called it quasi-equivalent. Head to tail, tail to tail, and head to head, for example. But the best way to remember it is just that there are pentamers and there are hexamers. So when any capsid has more than 60 subunits, that's when we have quasi-equivalence. And all that means is that the bonding environments are similar but not identical. But obviously, overall, they're arranged with icosahedral symmetry, and lots of the principles are similar. So this brings in the triangulation number, which is a useful number. And this is the number of subunits comprising the structural unit. So remember, the subunit was the lowest level. It's the protein chain. And then the structural unit is the next one up. So the T number tells you the number of protein subunits comprising the structural unit. So for a T equals 1, 1 subunit makes up the structural unit. For a T equals 3, 3 subunits make up the structural unit, and so on. T equals 4, T equals 13, and the number 60T will tell you the total number of subunits in the particle. And so as the T number gets bigger, the particle gets bigger, more numbers of subunits per structural unit. Anything greater than T equals 1 has a 6-fold axis of symmetry. So only T equals 1 is the only one that fits the Watson and Crick equivalence for everything in the capsid. Anything greater than T equals 1 it's quasi-equivalent, okay? You can build these particles with magnetic balls also. I was visiting a professor a couple of years ago, and he had them on his shelf. I said, I've been wanting to build those for years, so he taught me right there how to build them. And they're cool, because, and I made a video, which you should check out, building it. It's cool because you can make pentamers and hexamers with different colored magnets. And then you build the particle, and you can see what's going on. So here's a T equals 1 with 60 subunits. All the interactions are the same. I should have really made that with one color, but I didn't. And here's a good one to illustrate 180 subunits, T equals 3, where we have pentamers and hexamers comprising it. In theory, I could make really big ones. But I haven't, as I said, I haven't done it yet. So here's an example of a T equals 3. This is poliovirus. 60. So the structural unit composed of 3 subunits, but they're different proteins. So we call it pseudo T equals 3. Not important, but you'll see that. If they were all VP1, then we would not call it pseudo T equals 3, but they're different proteins. So it's a T equals 3, pseudo T equals 3. And so again, you have 5-fold axis of symmetry and a 6-fold axis of symmetry. And 3 subunits making up the structural unit, and that repeated to form the virus capsid. So that's a T equals 3 particle. Getting even larger, polyomavirus, SV40. This is a virus that will visit quite a bit. This is 72 pentamers of VP1. In other words, here's a pentamer. It's composed of 5 copies of VP1, so the same protein. That is, the VP1 is the subunit, and the structural unit is 5 copies of VP1. Here's how you build the virus particle. And again, you have pentamers and hexamers. Here is a VP1 pentamer with 5 neighbors, 1, 2, 3, 4, 5. And this would be a VP1 pentamer with 6 neighbors, 1, 2, 3, 4, 5, 6. So it's a little confusing because the structural unit is a pentamer. And then the pentamer can be found in 5-fold and 6-fold environments. Okay, that's where the confusion occurs. But this is cool because it illustrates how structure works. So you can see the N-termini of this protein are sticking out. And that's because they wrap into the neighboring structural unit to give it stability. So now here's an image that I've colored where most of the pentamers are purple. And then the one in the middle, I've colored each VP1 subunit a different color. And you can see this one is yellow. The N-terminus is snaking into the neighboring pentamer. That locks it in to give it some stability, right? And each of those does that. The red, the cyan, the magenta, the green, every one of these do that. So that's what gives it its great stability. Okay. Which of the following are characteristics of icosahedral symmetry in viral capsids? A, produces a solid with 20 faces, each an equilateral triangle. B, allows formation of a closed shell with 60 identical subunits. C, 5-fold, 3-fold, and 2-fold axes of symmetry. D, the T number describes the number of protein subunits per structural unit. All of the above. All right, how did we do? Most, it's all of the above. Every one of those is correct. A solid with 20 faces, each an equilateral triangle. You can make a closed shell with 60 identical subunits, 5 3-fold, 2-fold axes, and the T number is the number of protein subunits per structural unit. All right, we're going to make bigger virus particles. Adenoviruses, 150 nanometers. This is a T equals 25 capsid. It's made up of 720 copies of protein 2. So here's a trimer of protein 2. It's forming what's called a hexon. And here's an electron micrograph of the adenovirus. And most of those subunits are hexons. At the five-fold axis of symmetry, however, there is what's called a penton, and that is characterized by these fibers sticking out. And as we'll see next time, those fibers interact with the cell receptor. Here's the structure of the fiber at the right here. It is a trimer with a penton base and a knob that interacts with the receptor. So T equals 25, fibers at all 12 vertices. And in these bigger capsids, there are usually proteins with specialized roles. What do I mean by that? Well, these fibers are specialized roles, right? They're dedicated to binding to the cell receptor. But also, there are other proteins here. I'm going to point out one called protein 9, which is right here. It's this brown protein that is stuck in between these hexons. It's also called cement because that's what it does. There's a mix of pentons and hexons in this capsid, and that weakens it because it's not perfectly symmetrical. The interactions are not all identical. So when you have these mismatches, you lose a little bit of stability. And so these specialized proteins have come in to play some role in helping make the capsid stable. And in addition, there are also other specialized proteins here involved in DNA packaging and so forth, which we'll talk about later. So that's adenovirus. This is reovirus, which we talked about briefly. These are cool because they have two icosahedral shells. You have an outer shell and an inner shell. So reovirus is T equals 13, two concentric shells. So the outer particle is a T equals 13, and the inner particle is a T equals 2. So I will show you the outer particle here. It's dark. It's dark brown there. And in addition, it includes these light brown plugs. These green structures are called turrets, right? Turrets. And eventually the RNA is going to come out of those. But in the double-shelled particle, they're plugged so the RNA won't come out. And then there's the outer shell on it. And so we're going to see why that has two shells next time when we talk about uncoding. So that's reovirus and something unusual, a double shell. And then, of course, our bacteriophages, steampunk viruses, which are composed of multiple symmetries, right? In these particular tailed phages, we have an icosahedral head, which is built with the same principles that we've been talking about all along, their vertices and so forth. And then there is a contractile tail, which is helical in nature, and then long tail fibers, and a base plate. So the tail is attached at one of the 12 five-fold vertices of the capsid, right? This is icosahedral, and this is basically a complex rod like tobacco mosaic virus. Some of these can contract, and some of them do not contract. And at the base, so these little tail fibers are often used to help the virus attach to the bacteria. And then at the bottom is a base plate. And the idea is that the DNA, of course, is stored in the icosahedral head. And when this virus attaches to the correct cell, the DNA comes out of the head through the tail and into the cell. And sometimes that's accompanied by a contraction of the tail, but not always. The tail is the base plate is very interesting. And this is the structure of the base plate for one of these bacteriophages. So here in the A is the overall picture, the icosahedral head, the tail, and the base plate. And there's essentially a spike in the middle of this base plate that punches a hole in the bacterium. And you can see that illustrated here in red. The idea is once the particle is attached, the spike punches a hole through and then it falls away so that the DNA can come out. And in this particular spike structure, you can see, so here is the spike, and in the middle, panel C is the actual structure of the spike. It's a trimer of 3 different proteins, green, blue, and red. And if you take away the space-filling view and just look at the trace, the alpha carbon trace, you can see that the 3 chains are held together by an iron atom right there near the base. So it's tapered, right? It's made of beta sheets at the top, and they get narrower and narrower, just like you would make a spike, right? Except a human didn't make this. This was done by evolution. So a spike is exactly how you do to punch a hole in the membrane. So for that reason, I think this is really remarkable. And again, the spike falls off once it punches the hole. The DNA is under huge pressure, hundreds of pounds per square inch of pressure, 600, 700, 800 pounds per square inch. You know what your car tire is? Like 32. This just makes fun of it. 700 pounds per square inch. Try inflating your bicycle to 700. No, don't do it. Don't do it. It'll explode in your face. But it's really remarkable. And there are motors that get the DNA packaged in there under that high pressure. Okay, and then we have herpes simplex virus, another one of these complicated capsids. So this one is a nucleocapsid within an envelope. This is the actual cryo-EM structure of the nucleocapsid on the right. And the cool thing about this nucleocapsid is that, it has 12 five-fold axes of symmetry, right? One of them has a portal. And the portal, you can see, well, this is the structure of the portal here in purple. You can see it on the actual capsid here. There's one portal per capsid, no more. And the portal is where the DNA goes in during assembly and where the DNA comes out during infection. And this is, again, the DNA is packaged in these particles at really high pressure. So once the portal docks onto the nuclear pore, the DNA comes shooting out. Very cool. Anybody ever play that game Portal? Yeah, this reminds me of Portal. Yeah. Okay, many people have given me herpes virus keychains. I don't need any more. I have plenty, thank you. But this is actually very cool. So here's the keychain. It's the herpes virus, the outer envelope, right? Then you can open it, and you can see the capsid here, right? And there's the portal. They actually have a portal. It's very exciting. And then you can open the capsid and see the DNA. So this is a multi-layered keychain. Very cool. I'm amazed at the accuracy for this, right? Now, capsids can be covered in membranes. As I've mentioned before, these are derived from the host cell because the virus does not make lipids. These are typically acquired by budding. They can be acquired at the plasma membrane, at the nuclear membrane, at the ER. And the nucleocapsid inside can be helical or icosahedral. And when you have a membrane, you have glycoproteins embedded in the membrane because that's how the virus particle can recognize the cell. These are called integral membrane proteins. They have typically an ectodomain, a transmembrane part, and a part that's in the interior of the particle. And often they are multimers. They form what are called spikes when we look at them under EM. And these glycoproteins can be perpendicular to the virus membrane, like the influenza virus HA. They can be parallel to the membrane like the flavivirus E protein. And we'll talk about how these would work next time. And again, inside these envelopes, you have nucleocapsids. They can be helical, right? RNA protein complexes, or they can be icosahedral. Doesn't matter. So you can take a helical or an icosahedral particle, put a membrane on it, it's now a nucleocapsid. There are other things in particles besides structural proteins, by the way. Here are our third category of viruses. Complex particles that bear no symmetry, as far as we can see. Poxviruses, Pandoraviruses, Pithoviruses. We recognize they have membranes, they have pores through which nucleic acids can pass, but we really don't understand very much about how they are built. Okay, and besides all the structural proteins, there are other things in virus particles. There are all kinds of enzymes that are needed at different parts of the reproduction cycle, and often cellular components as well. So it's not just the viral structural proteins, it's other particles, other proteins that are picked up as well. All right, now you know how to build a virus particle. Next time we'll show you how to get it to attach to a cell and liberate its nucleic acid.